All right, so is there anything else? So as Rachel said earlier, if you have questions, feel free to write them in the chat and she will unmute and she will stop me as I go. So you don't have to wait till the end. I'm happy to answer questions, but if you want to wait till the end, that's okay too. All righty, so let me just figure out how to get this going. Does anybody have any questions right now? No.
No.
Cool. So give me 1 second to screen share. All right, can you guys see that?
Yes.
Okay, so it's fun to present on Zoom because it's less nerve-wracking, but it's also kind of weird. So I know you guys are all there and I'm happy you're here. So, all right, so Citizen Science 101, how to document fungal biodiversity. So we're going to be covering a range of topics, including just an overview of citizen science, how to photograph and collect fungi for scientific purposes, navigating a website called iNaturalist, and uploading your observations, as well as collecting and dehydrating specimens for potential genetic sequencing through the Fungal Diversity Survey. So what is citizen or community science? It's also called civic science, whatever feels good. And this is just kind of an extension of our thoughts of traditional science. And it's scientific research conducted in whole or in part by amateur scientists. And so I know in like our common language, the word amateur kind of gets looked at as like, oh, it's non-professional and it's kind of someone who does something on the side, but the root is actually the same root as amor, so love. So it's somebody who does something because they love it. And I've heard this a few times, and I love to remind people of that because you don't need to feel like you're not important or that you're not a professional, because there's a lot of ways to be a scientist as a community member. So the goals and outcomes of this initiative is just simply to advance scientific research by improving the capacity and the accessibility of scientific communities, and also increasing the public's understanding of science as well as their involvement. And our club is really involved in citizen science. And I'm gonna show you this kind of hopefully not overwhelming slide. So this is a pretty big project that started off as the North American Mycoflora Project, I think in 2017 or so, and it's now called the Fungal Diversity Survey. So those are it's the same project, just new name if you see that anywhere. And so the goal is to equip citizen and professional scientists with the tools to document the diversity and distribution of fungi across North America. That's their direct quote. And there's a lot of ways to do this I'm going to go into, but our club to start has 5 projects that have been associated with this greater project, and it is the South Sound Fungal Diversity Survey, which is just kind of all macrofungi that people have been finding. This is like where most of the specimens have gone in through, and it's the biggest project that we've had for our club. And then we have Hygrothy of the Lower Sound, which is their big project that has had a lot of exciting findings. And these are the waxy caps. So this is the family that includes the many genera of waxy caps. And we have Olympic polypores and Romaria of Western Washington. And the newest project that I've helped start with Luca Hickey is Entomophoreaceae of Western North America. So it's a pretty big project. It's just started, and that is the pink gill mushrooms. So Entoloma, Leptonia, Antosophy, etc. So the general goal of this project, at least in data, is to get sequencing. So we find the mushrooms and we take tissue samples from them and we send them in for sequencing. So we send them to a lab, they undergo DNA extractions, PCR, which is polymerase chain reaction, where we just amplify the DNA, and then the ITS and maybe LSU barcode sequencing. So this is ribosomal DNA, just as part of this long genome of the mushroom. And we kind of have this barcode that has now been standardized where we go in, there's molecular technology that kind of goes and finds this part and amplifies it. And this is where the most variation between species occurs, because a lot of the genomes are just kind of the same bits. And so we can see differences between species by looking at this specific part of the mushroom's genetic code. And we can then take the sequence and we can run it into a online database called, they go into as entries on GenBank. And this is an international database that has all organisms on it. And then we use this program called BLAST. I'm using a lot of technical terms. I'm going to get past this. This is just kind of the overview of it. But BLAST lets us compare our sequence with everything else. So we could have put it in as one thing and it could come back as something completely different, or it might match exactly. And so, yeah, they can be confirmed or challenged. And this is a code, genetic code sequence that we got back on the bottom here. And this is one of my waxy caps that didn't match anything. And so, it just looks like gibberish until you start seeing all these things pop up from all over the place. And it's really exciting. And it's a process that takes time to understand. But this is just the general goal, the end goal of it, besides the documentation process. So why do we do it? And this is a phylogenetic tree and it looks terrifying. And I know that. And so I just wanted to show kind of a comparison of, yes, we want to contribute data to science that can help us fuel research, ecology, conservation efforts. And it ultimately helps us to group fungi with other species or greater groups that they're related to. So when we were classifying, fungi, when people were doing this for hundreds of years, it was based on what they looked like. Then it switched over to what their spores looked like. Kind of, we got into microscopy. And now we're in this molecular age where we're able to actually compare their genetic sequence and see what's what. And so there's kind of a ripple through taxonomy that people get really frustrated with. But it is really important that we do this because it helps us better understand them, which we can apply to so many different parts of science. And then these pictures on the right are just to kind of show. It also helps us to appreciate biodiversity and just the abundance of species and groups of mushrooms out there that, we think that there's, 2 of one and there's 10 or whatnot. So they're constantly, we get these results and it's very humbling and exciting to see how much is out there. And, just for the sake of better understanding these organisms and giving them the attention that they deserve. So with the technology that we have available to us. So here's just a little smorgasbord showcase of some of the beautiful waxy caps that we have collected and either have gone in for sequencing or are awaiting sequencing. And honestly, that could be its own entire project because we've had some really incredible results from all of the mycoflora projects. Fungal Diversity projects. Still got to train myself here. But so I just thought I'd show you guys a few of them. And so the one up on the left, this beautiful blue parrot mushroom, Luca found, and we look a lot at the parrots. There's a bunch of them. They're usually typically green and yellow, but there's at least several species that haven't been, separated from this one group, from this one species that they're known as. And this is one of them, and it really doesn't have a lot of information out there on it. There have been a lot of collections of it, and it's probably going to come back pretty different, but it's very beautiful, and we were excited to find it. Then Hygrossa virescens, the lime green waxy cap. This is just such a stunning mushroom that is very elusive and only fruits. It can go decades without fruiting and then suddenly fruit all at once in different areas. And there's just been mycologists who have, seen it once in their life or not at all. And so it is rare, but also abundant when fruiting. So it's just a fascinating one that we're excited to sequence. And then on the right is Hygrossa latissima. And this mushroom was thought only to be, the species was thought to be in California only. And then I found it at our college campus, and it was the first confirmation, on paper that it was growing outside of California. So I got to write up a little entry. Regina helped me put it on the Burke Herbarium site, and they used my photo. I had wished that I took a little bit better of a photo, but it was really exciting to kind of be part of that. And this is like the initiatives and things that, can happen. I wasn't expecting that, and unexpected things like this happen all the time. And then the devil stool, Eric and Jen Chandler's prized find, that was really, really exciting. And it was kind of a global little phenomenon. And it was a lot of communication with mycologists all over the world, initiated by our very own club members, because they found this rare mushroom that was growing in a grassy area in Lakewood at the technical college. And if you see it, you got to let them know. There's wanted posters we have that we can show you. But it had only matched, I think, one in Europe until we had one sent in from Canada. And so it's probably needs to be redescribed, and it's really just been super exciting to see that all go down. And I don't even fully understand all that's happened because it's been a lot. But then we have the Chromacera in the middle, and it's so Chromacera cyanophylla was the one that we had for the area in the books, and we had been at the Key Council down in Southern Washington. Sorry, I have loud neighbors, so sorry if you hear them. So this one we just assumed was that one. Went in for sequencing, and it came back that it's different than this one, and this specimen is being part of the process of rewriting it as a new species for Western North America. So we're waiting on this paper to come out so we can tell you guys what it's called, but it's a beautiful tiny mushroom growing on wood. And then that is my little undescribed hygrosity species that didn't match anything in GenBank. And that doesn't fully mean that it's undescribed, but typically the species that we have down have been sequenced at this point. So, but sometimes wax sea caps, the type specimen can't be sequenced, and so you never really know. And a big shout out to Steve. As Rachel said, he's done so much for this project, all the projects, and kind of helps keep all the gears turning. And I could make a whole slide of all the amazing finds that he's sent in, because they come back really interesting too. All right, so iNaturalist is a fabulous website and platform that I'm going to really push on you guys to get on. And it's actually crucial for you to use it if you want to be involved in the Fungal Diversity Survey. And it's a way of mapping and sharing observations of biodiversity around the globe. This is their words. So it's a website used by naturalists, scientists, or anybody who wants to identify any living thing that they found. So it's not just for mushrooms, birds, fish, algae, you name it, you can put it up there. And so it uses a combination of artificial intelligence and community source identifications. And it's a really cool combination that it takes a little time to figure out the process, but I'll show you how it works in a little bit. And it's so much fun to use once you get the hang of it. And like I said, it's the social media platform of your dreams, if you're a nerd, you know? And so this is also part of their goal, but is providing valuable open data to scientific research projects, conservation agencies, and other organizations, as well as the public. And it also has an app that you can use. And I loved this little graphic I found of somebody from iNat made. And I had to put the little mushroom in there because there was no mushroom. But like I said, you can photograph anything. You can do it on your camera or your phone, and you're going to upload your pictures. And I'll show you how to do that. Uh-oh. Okay. So this is a crazy, just beautifully lit up map of observations from the world. And this is all the observations on iNaturalist. So 56 million plus, and added really. And there's about 1.5 million close to observers, and going to be growing now that you guys are going to get on there. And so this is an interactive map, and I'll show you how to use it. There's so many great ways to filter different things based on what you're interested in. And this is just like kind of including everything here. And then we have fungi, including lichens, which makes up about 6% of the observations on iNat. And still pretty much a lot of parts of the world, you know, we're missing some desert mushrooms here, but doesn't mean they're not there. Okay, so how do I use iNat? So I'm going to try this little interactive way. I'm going to pull up the site and I'm going to show you how to do a few things here. Because a lot of people, I tell people about iNat a lot, but I feel like there's some quirks, but it's gotten really, really easy to use. And I feel like if you see it in action, you'll be more motivated to get on. So here we go. So I've already logged in. So this is my home page, and it's really very much like social media in the way that there's like a little home page here. And this includes observations from both me and other people. This is just like updates, like people who are adding identifications or adding observations. And you can click on it, where if you want it to just be stuff that you found with like little notifications, you can get that as well. this mushroom is popping out a lot. And so let me just see real quick here what I want to show you. All right, so explore. So this is something that if you don't want to make an account, if you go to the main page, it will say explore or log in. So this is something that anybody can do. So it's just really fun, I think. And I want somebody to, unmute and give me a mushroom or a type of any scale of mushroom that you want me to look at on the map here. Species, genus, family, whatever you want.
Romaria.
All right, got it. Oh, we got a lot of Romaria lovers here. Okay, so I type this in and it's like, do you want to look at a specific species? Do you want to look at the family that Romaria is in, et cetera, et cetera. So it's kind of depending on what you want to look at. We're going to look at the genus. And now this is narrowing all of the observations to just this genus of coral fungi here. And so now somebody give me, I guess we should do, I was gonna say, give me anywhere in the world, but I guess we should do, give me some, ooh, hold on, I zoomed in. Somebody give me a location, relevant, I guess. Where? Did I get a location?
Mountains.
Mountains?
Olympic mountains.
Sorry, I didn't hear that.
Olympic mountains.
Oh, Olympic Mountains. Okay. Let's see if that's a location on here. I'm gonna put in Olympic Peninsula because I think that's an option. No, it's not. Oh, maybe. Let's see what happens. 0. Okay, well, there's 0 here because it's a very small box. Let's say Quinault. Okay, so there's 8 species here, 3 identifiers and 5 observers. So 5 people have reported up, including Luca, who is always finding lots of stuff. Let me make it a little bit wider. Let me just say Washington, just for the sake of it. And you can change, you can make it interactive. If you move, you can say search, update, map, search. So here's all of Washington, clearly more concentrated on the west side. And so this is just helpful to visualize different groups that you are curious about or just kind of anything. And a great way that I like to use this is I kind of just look at what people post in my neighborhood. And that's something I did when I first got on iNat that really helped me to, learn what I was looking at when I was going around. And it's just cool to see, people in your neighborhood who are finding tons of interesting things. And sometimes, you can help them with their observations and they can help you. And I'll show you how to do that more. So I'm going to exit the Explorer. Let's see, now we're going to go back. Oh my goodness. Thank you so much for just following this with me. All right, so now I think I'll show you how to make an observation. So upload over here. So you're going to upload your photos. And so this is the desktop version. The app version, it's a lot of people take photos on their phone. So it's going to be a lot easier for you to upload photos directly from your phone on the app. And that's one of the pros of having the app. I recommend it for that. And you can do a good amount of the stuff that you can do on the website on the app, but the website's just, I would recommend playing with both because there's some limitations on the app. But I take photos on a little camera and I upload them to my computer, so it's easier for me to just use the desktop anyways. So I have so many things here. All right, so I've cleared this, ready to go. So we're gonna make 2 observations. I'm gonna make my 1000th observation with you guys. Should definitely have a lot more by now, but it takes time, to upload pictures. So I'm dragging them all in. And this definitely looks like more than 2 things here. And I'm going to show you how to avoid accidentally making more observations than you intend to. I think I have one more picture here that didn't work. Let me see this one. And there's a limit on how many you can upload, but I think it's 8. It's different on the app versus the site, but you can upload several images for one observation. And that's just to try to get different shots, different angles, and I'll go over photography of that in a little bit. So what I'm going to do here is show you how to just combine these because, excuse me, this is a big problem that I've seen with people who are new to iNat is that they go to upload their pictures and then they have 4 different observations for the same, they intended it to be one, but they uploaded the photos, they came separately. And that's easy to do because they just show up like this on their own. And what you have to do is drag them like that. And now that's one. And so this is just a really important thing to know how to do if you're using the website. And so you can pick which one you want to be the first photo. And I would say probably the most indicative of the species for that one, you know, the one that looks most like this one, I would say probably for this one, but it doesn't really matter. It's your choice. And so if you pick that one, you have to just start dragging the other photos to that one. And on the app, there's a little checkbox that says, like, I want this one to be, I think it says cover photo or something. It's been a while since I've been on the app. And so look at this cute little baby. So sweet. It was very tiny. This was during one of our mushroom club walks at Evergreen. I was with Amy and Andre, who are actually on this talk right now. So here are my 2 observations. And so there's no information besides the date, and the time doesn't seem right, but it's not very crucial to have the time. So what I'm going to show you is how the artificial intelligence works, which if you click here, well, actually, does anybody want to shout out about this one? Does anybody know what this is here? No pressure. This is a very common Pacific Northwest mushroom species.
Lauren, here's a question. Are those phone photos, or do you have a macro lens on a DSLR?
It's actually neither. It is not a phone. It is a small little point and shoot camera that is a macro geared camera. It's Olympus TG6 Rough. It's supposed to be waterproof. It's submergible, but I haven't done that, and I don't know if I would, but it's just small and tiny, and it's great for macro photography. And so definitely it's more affordable and easy to carry than a DSLR, and the quality is better than a phone for sure. But you definitely don't need those types of cameras to take good mushroom photos. And I will give you some tips on that in a little bit.
Somebody suggests that that's Stropharia ambigua.
Oops. Yep, that's right. So see, okay, so that's down here. So if you click on this, and the AI works differently when it has the location. So another thing that's tricky about the desktop is it depends on if you use a geo-tagging device. So this one, sometimes it is on this camera, sometimes it's not. I haven't fully figured that out, but I'm gonna go, I know where I found it. So this is something that you need to note because you do need the location to be on there. So if you're taking photos that aren't on your phone, or if you're using your phone and you have your geolocation tagging off, then you're gonna need to mark where you were when you found it. And you can keep it kind of broad. And if you found something at your house and you don't want people to find it, you can obscure the location, which means it'll just kind of toss it like a mile away or something. And that's an option for privacy. So you can type stuff in. So I'm going to type in Evergreen. And I was right about here. See, this one's like, I think I was like right there. So, and you can make, you know, you can make this really tiny and really exact. But if you were like, all I remember is I was at Evergreen, you can just kind of make it huge and it's totally fine. Or you could make it, I don't know how broad you can go, but you can do like Olympia probably if you wanted. So I'm just going to put that one there. And then a nifty thing that this will do is if you're uploading on the desktop and you're doing multiple from the same day, which I recommend doing batches of the same day, because you're usually at the same location, and it will remember and it will say, were you here with the other one? And I was actually, I think, I was a little bit further away for this one. We're going to go all the way over here. It's fun to drag around the map, but you can do this a variety of different ways. So I was right down over here. So there's that. And it really doesn't have to be precise. As long as you have a location that's as close as possible, it's better than nothing. And that's the important part. So this is how the AI works. So if you just tap on this, it's going to load suggestions based on my photo. And if you have the location in, it's going to combine both visually similar as well as seen nearby. And so this is not what it is. And like, am I wrong here? I'm like questioning this common mushroom.
Well, it had the Stropharia and
I know. Yeah, I totally did. I'm just like,
oh,
So here's Stropharia ambigua down here. And it's not the first one. So you can't just expect this technology to give you the answer right off the bat. A lot of times, and it actually depends on the photo you use too. I've noticed that the algorithm will change a little bit depending on which is the primary photo. I had it suggest like a frog when I was doing a slime mold earlier because the pattern looked similar to this frog that it was trying to compare it with. So it is correct here, but it's not the first one. So this is a great way to practice your identification and kind of have like a little robot as your mentor because if it just gave you the right answer right away, where would be your motivation to learn? But a great thing that I do a lot of the time is say I'm completely lost. I have no idea what something is. And I've looked in the books as close as I could get. But before I knew how to navigate the books and check certain genera, I would just be like, I don't know where to start. And so I would upload this photo and then I would start referencing the suggestions. So I would say, okay, let me look at Satharella. And I would go look up Satharella in the book. And so it's kind of a great way to pair like technology and traditional references that are more concrete. And so my recommendation for this is to not click on it as an ID unless you have some kind of way of generally knowing that it's that specific thing. And so if you're like, well, what if I don't know at all? So if I had no idea what this was, I could just say fungi, including lichens. Bam. And now this is just grouped into the whole kingdom. And it's going to be grouped in with a lot of other things. And so if you knew what the genus was, you could say, I know it's maybe a Stropharia, but I don't know what Stropharia it is. You could say Strophareaceae as a family, or you could say genus Stropharia, roundheads. I've never heard that. iNat has great common names that are pretty funny. So you can just click on that. And then the way that the community-based identification support comes is that you'll upload it and then people will interact. People who have accounts will go on the identification page or mess around on the map and they will confirm your identification or suggest something different. And it's not a guarantee that they will, but the more you use it, the more likely that is to happen. And the more people on iNat, the more identifiers there will be. And so don't be discouraged if you upload something and nobody confirms it. But if a few people, I think it's 2 or 3 people confirm the ID, it becomes research grade, which makes it kind of a more viable observation in terms of being reliable to that species. So I could talk about this forever. So we're going to say Stropharia ambigua, but if you aren't sure what it is and you have a guess on the genus or the family, get as specific as you feel confident for. And that's always my advice with this. So kind of have to meet it in the middle. And then you could say there's a notes thing here. So you could say.
So Eric says there's a view button that gives you a close-up of the photo from the listed possible for comparison.
Oh yeah, that's a good thing. So you can click this. I always click like view it in another tab because it opens it up on the page. So this is a great, thank you, Eric, because this is a great feature of iNat that look, top observer is our very own Luca Hickey. And so this is a great page that every species and every, taxonomic group has. And it shows you on the map where it's been found. You can click about and you can read about it. There's not a lot of information on this one. Sometimes they have a lot of info, sometimes they have less. This is sourced from the Wikipedia. You can make the Wikipedia page if it's not there. And so it shows you the taxonomy too. So if you want to learn more about like different groups of fungi, how they're related, you can kind of start here, like with the ones that you're finding, you can see how it's broken down. And this is going to help you over time. This isn't like something you're going to want to jump right into. You can see if it's endangered anywhere. You can look at similar species. So this is a great way to explore ones that are commonly misidentified as that species. So, and it shows you a little bit of the phenology, so the seasonality of fruiting and whatnot, and there's a little leaderboard here, and it's really a lot of fun. So and it's really educational. So we're gonna go with that. I gotta kind of make sure I'm picking up the pace here. And so this is the lichen agaric, and this is absolutely right. So cheers to the AI. So we're gonna click on that, And there's a lot of mushrooms that kind of look like this. So this is actually a lichen, you know, it's actually kind of crazy, but a lot of our lichens are formed with ascomycete fungi. So they're the cup fungi. And when they fruit, they often resemble those kinds of, you know, little cups. But it's a waxy cap. It's in the waxy cap family, but it has a lichen association. So this green bit on the bottom here is the algal mat called thallus. And then we have this beautiful little mushroom popping out to spread sexual spores. So that's a really fun one. And if you wanted to say like on decaying wood, it's really great to reference the substrate. If you know, it's good to have pictures of it, but definitely reaffirm it in the comments if you in the notes if you can, and mention if you smell it and it smells like bubblegum, or if it smells like tomatoes or mothballs, you can say strong odor of this, and that's going to help people confirm or change your identification. So definitely give as much info as you can. And so I'm going to show you how to add it to a project, but so you can add things to projects, which I'll go over in a second. You can do that either on here while you're uploading. This is the individual observation I'm clicking on that's changing this sideboard. So if I wanted to put this into the South Sound Fungal Diversity, so these are some of the projects I've contributed to, and I'm going to type that in. And because I've joined the project, I'm able to directly put these in, and I will just show you how to get to the page so you can join it and do that.
Here's a question.
Yes.
It says if you post an observation labeling what you think it is, but someone posts another possibility, what kinds of questions should you ask the person who posted something different to help verify the identity of the fungus?
That's a great question. And the people anybody could go on and suggest identifications, just like anybody could make observations. So it's going to vary depending on who is doing the identification for you. And a lot of the people who make observations are really nice. They are maybe scientists or just amateur mycologists who love to help other people. So what I would say to do is there's a comment section, and that's where in the comment section, it's going to show you that somebody suggested a different species. And what I try to do is write a little snippet of why I think it's that versus the other thing, so that the person can make that association. But if they didn't and they just said, I think it's this, you can tag them. So they'll have their username. You just use the @ symbol like you would on other social media. And you'll see their name right there when they suggested the name. And you can say, you know, @Lauren Ray, what made you think it was this? And I did that actually the other day for a liverwort. Somebody was telling me it was a subspecies, and they gave me a link to a paper that was explaining it. So someone might give you some information like that, or you might never hear from them again. And so this is you can't expect anything, 100% on this. And it's going to take, the more observations you make, the more you're going to interact with people who are willing to help you and answer your questions. But there's no guarantee. And you can ask, I would just say, like, what features are distinguishable for that species. And you can also click on the species they suggested and read that about page. And you can also compare. Something I do a lot is I compare other observations of a species that either somebody suggested to me or that I'm considering putting, so I can see if I'm in the right ballpark. And, you know, that's a way to go. So move here. So here's that says submit. So here I am going to submit these 2 observations. You just got to make sure you have the best name you can give and the date and location. So here we are. Here's all my observations at Evergreen. So this is a list of all my observations here. There's lots of ways to look at your observations. I really can't go into all of it right now, but if I wanted to look at just my own, if I click Europe, let me see, where's the map? Like I'm saying, you got to toggle with it. So here's a map just showing my observations, and I got 1,000 now. Woohoo! 475 species. And so these are the filters I just want to mention real quick, is when you're on that map, you can not only search for specific species and locations, but you can search who posted it. You can search just a kingdom of life. You can search a date. You can search it through a project. If it's research grade, which means it's been had several people contribute to it, and lots to do there. Okay, so let me just see. Last thing I'm going to show you is just how to join a project. Nice and simple. So if I was not part of the project for the South Sound page, I would type in South Sound Fungal Diversity Survey, and there it is. So if you click view observations, it's just going to bring this map up and just show you what people have found. I guess I'll just open that and I'll show you. So if you click on that, I do this all the time where I mean to click one instead of the other. So like I said, you really try not to get frustrated, just mess around with it. So here's the one that I just added here, and we have 807 observations for this project, and they're all over here in the corner. Oh goodness, I accidentally zoomed in. All right, so yeah, here's all the observations that people have made, and then here is the project page. So I made this iNat project, so it's asking me if I want to edit it, but this all the way up in the corner, for this style of the traditional project, there are several kinds of projects. This type requires you to join the project. So it's going to say join project here, and you can join that. And here's some of the people who make observations and species, and here's the map. And then there's some information. Here's the link to the Fungal Diversity Survey, etc. So this is where stuff goes in, and then not everything here gets sequenced, but this is kind of like the choosing board where like things go where like if I'm not sure I'm going to send something in, I add it to this project as like a potential. So if you find it's something that you are maybe considering sending in for sequencing, now you'll know how to join the project. And then last part of adding an observation to a project, because like I said, you could do it when you make the observation, but if say you had one already that you've already made and then you just joined the project today, and then you're like, I want to get the sequence, I already have it dried, definitely not going to do a snake, say it was the turkey tail, you can click on your observation and it's going to load. So here's all the photos that I had combined. Now you can, this is how the view looks, where you're like, oh, okay, I see the, all these different angles of this. You can favorite observations that you think are interesting. And here's it waiting for, oh, look, see somebody, I just uploaded this and somebody suggested the ID. So this is helping it improve. And it's automatically added to this project. But if we want to add it to the South Sound one, you have to manually add it in. So you either do that upon uploading it or do it here. And I have to stress that because other projects sometimes just pull them and put them right in, but we have it where you have to choose to add it. So now it's here in the project page, and all I did was search it and then it adds it in. So that's it. Now I'm gonna stop doing this. Is there any questions so far?
So far, people have been answering each other's. So far, so good. And I'm interrupting when it looks like it's a question that others aren't helping with.
Cool, and I'm definitely, it's 8, so definitely going past the time. And so we are recording this just as a reminder. And if you need to leave, we are happy to share the video with you. So this is a lot of important stuff, and I want to make sure to be thorough and kind of cover it all, because we want you guys to contribute, and a lot of people are curious about this, and so I'm gonna just get through it together. All right, so photography is really important for scientific documentation of any organism, and people who love mushrooms often just love to photograph mushrooms, and so this is not like a requirement for you when you just want to casually take pictures of a mushroom. Be artistic by all means, but this is going to be really helpful for iNaturalist, which even if you don't send stuff in for sequencing, the process of adding stuff on iNat is going to help you get better with your ID. And so in order to identify stuff, we need to see different parts of the mushroom. And that's pretty crucial because, and I'll go over a little bit more why, but these are the goals when we are photographing mushrooms. We want the image to be clear and we want it to be in focus. And so I was like, not sure if this was like the same thing, but I think the way to distinguish it is that we obviously don't want the overall image to be blurry or fuzzy or kind of like, when you're moving and it's like streaky, we don't want that. And in focus, meaning the mushroom that is the subject of your photo that you are trying to make, an observation.
Well,
yes.
Quick question. Somebody's asking for the name of the camera again.
Oh, okay. Yes, let me it's Olympus. I'm trying to see if I can get to the chat. I use Zoom every day and I can't. So Olympus, Olympus, Olympus is the brand. Rough TG6. And I can write that in the chat when I can find it again.
There, somebody just wrote that in. Somebody just
Great,
wrote it
thank
in.
you.
And then another question, so sorry, for putting it into the South Sound Fungal Diversity Project. If somebody's in Spokane and they find they've got, they've got findings, could they put their findings in?
Yeah. We've had, yes, they can. And they should definitely add it in. We've had stuff come in from a variety of different places, sometimes technically through the project, and we've also sent batches just in through the club. So we kind of have to talk about it because I'm not sure how strict those parameters are, but I'm pretty sure that they're not. And I'm sure Steve, if he has an opinion at the end, can help me with that. But I would say yes. There's also, I'll send you a link to other fungal diversity surveys. So there might be one there as well. And I would say that would be the priority, is to talk to whoever's doing that one. But I don't know if there is. So hang tight on that. Okay, so we were saying in focus. So you want the mushroom that you're photographing to be in focus, because sometimes I'll see a picture that's not blurry, but like the leaf of the plant is more clear in the photo than the mushroom is. So that's what that is trying to explain. And so in situ just means in place. And so you want ideally to have a picture of the mushroom in its habitat. And this is going to help us have reference on the mushroom, figuring out, is it growing on that thing or with these other organisms? And it's not required. You can still take great pictures of a mushroom at your house and probably get the identification if you know anything about where you found it. But it's ideal. These are the goals, not necessarily the requirements. And you also want several angles. And so that's what I'm showing in the right side here, the underside. And that's a huge one. That's the most important one, because you would not know if a mushroom has gills or pores or teeth if we don't look underneath. And so I felt pretty bad the first time I picked a mushroom, because I didn't know anything about what I was doing. And I hollered over, and I found one. And the club I was in Long Island, they were just like, pick it. And so I picked it, and that's how you learn what they are. You got to look underneath. You got to see what's going on. And so we've got by the side, it's kind of like, you can see the side in both the middle one here and the one on the left. The light kind of shows the texture of the stipe a little bit better on the left. And so just kind of any variety of getting the side is helpful. And then the top, just, and you can get that also in a variety of ways, but seeing the different angles of the mushroom is really, really helpful. And sufficient lighting just means that we can, it doesn't have to be perfect. Some people bring bits of tinfoil or like little ways to like bounce the light. And that's if you're getting, serious about it. But by all means, as long as, if you're using your phone, there's ways to tap on the screen to change the exposure. Because if you're in the dark woods out here, there's a lot of cover. It's really easy to get dark photos. So if you don't know how to change the light settings on your camera, then definitely figure that out, because as long as we can see the mushroom and the colors, then, and the shape of it, that would be good.
Certainly different ages of the mushroom also make differences in terms of, some of the mushrooms change dramatically as they age.
For sure. And I'll show that a little bit too. I didn't put the names of these mushrooms in, I don't know why, but this is Arenia chlorocyanea. And I had a sequence go in of this that I was on a field trip to one of the prairies out here. And my professor was like, they found blue mushrooms here a few years ago. And then I found it, and it was like, it's a really dark teal. It's beautiful. And it wasn't this exact one, even though it's the same species. And that one had undergone a name change. And my sequence was the first one in the database with the correct new name. So that was one of my first accomplishments. This is the midnight Entoloma medianox on the bottom. Very beautiful mushroom. Okay, so I'm not going to go into explaining what all these guys are, but my point in this slide is to show you that you can accomplish getting the different angles in one shot. You don't need to take 7 photos of every mushroom like I do, but you can if you want. But if you're just trying to be efficient and you want to get, the best photo you can get for your specimen, flip one over, pop it in with the ones that are still the way that they, were growing. And then you can just kind of achieve this one and done. And if, a lot of times if I'll do one like that, and then I'll take shots of it separately. So that works great. Or the second method I would say is just to, photo of kind of the top or the side. I like a mix between top and side like this one, and the underside of these gills here. And these are just beautiful mushrooms, but this is Marasmius placatulus. I said I wasn't going to say what they are, but I can't resist. The bottom is just the way that the cap repels the water, and the gradient on the stipe, they're just so beautiful. So, and then like Rachel was saying, these are just some additional things that you can photograph that are going to help you get identifications. And so distinguishing features like the, I'll say substrate on the right. I'm like, where do I start? This is a Lactarius, so it's the milk cap, and I scraped the gills with my finger because I knew it was a milk cap. If you don't know, you might not figure out it's exuding this milk until you start to handle it. And at that point, that's fine. Take a photo once you notice. I'm gonna just turn my heat on because I'm really cold. Hold on. I'm shivering. All right, so the milk caps are a great one to get, to show that there is a reaction happening, and there's bluing that will happen, or just staining of different colors. And so I would definitely say photograph it if you can. And this photo also shows the substrate. This mushroom is growing out of this old decaying log, or kind of like right from the bottom of it. And then on the white mushroom up on the top, this agaricus has a partial veil. So the gills are exposed on the right because I ripped off the veil, but they weren't open yet. So if you flip the mushroom under, all you see is just that white cottony fiber. But it's really great to get a shot of that because that helps us identify agaricus. So, and lots of other mushrooms as well. And I got both the gills, and the partial veil. And so the partial veil is intact here. The mushroom in the middle, the veil popped off, and then it becomes a veil remnant. Now I'm going into my morphology talk. All right, so growth stages are also great, and I was able to get them all with this honey mushroom just in this one shot. You could see the tiny baby in the bottom, and then the mushrooms that are getting a little bit bigger with age. And these ones also have a lot of different fibers on them, fibers on the cap, fibers on the stipe, that are helpful to see at different ages. And then scale. Oh, I don't think it says scale anymore. I can't see, but it used to say scale.
That does.
Okay. This is covering it. So it's not necessary to get scale. It's taken me a while to try to do that and remember to do it. But it's always helpful because if you get like a close-up of a mushroom, you might not know how big it is, and that can help you make an ID. So
somebody asks, would you ever want a photo of the hyphae or the mycelium?
It doesn't hurt. I'm one to get excessive photos rather than wonder. I get to the field guide and it's like, mycelium is purple, which happens sometimes, or like the basal hyphae at the bottom of the stipe will be like a lilac, or it'll be really thick and fuzzy. And so definitely, yes, if you can see it. The tricky thing is if you see it kind of close by, but it's not connected to the mushroom, you might not know that that's for that species. So if and they can all grow pretty close with each other. So if you can see it kind of at the base of the mushroom you picked, I'd say yes. Then we can assume it came from that specimen. But and you will see cool colors, and like honey mushrooms have like really, it's called shoestring fungus because of how black and thick these crazy like rhizomorphs, I think they're called, parts of the mycelium get. And it's a really aggressive pathogen, and that probably helps. So yeah, I'd say yes. Long way of answering questions. So expect muddy knees. There's, you really can't get good mushroom shots unless you get a little bit closer to the ground, especially if you want to get it in its habitat, because they're often growing. Unless you're getting a polypore right at breast height, then it's not going to be so simple. So these are our club members. There's me on the bottom, and our president Rachel on the bottom right. And this is Luca, and I don't know if he came tonight, but it's his birthday. Happy birthday, Luca. And he's the most committed to getting the best shots, and he will take the time he needs, and he will get as dirty as he needs to get these shots. And so I have tons of photos. I can make a photo album of just him laying on the ground. And then here's Steve up in the top left, getting a picture of our dragon egg amanita. And so, yeah, and you don't have to get filthy or dirty or roll around in the mud or anything, but you can just crouch down like Rachel's doing or I'm doing. But I mean, I get muddy knees every time I go into the woods, and it's because I'm taking pictures of mushrooms, and it's worth it, you know? And so just be prepared for that. Some people don't want to get dirty, but it's just part of the gig. Okay, so not too far from being done. So thanks for hanging in there, guys. All right, so here's some examples of bad shots. And the ones with the ... I'll tell you why ... But we'll start with the one on the right. And so this is clearly a blurry photo. It even says blurred background. So it seems like they made it blurry, but that's a very common occurrence, and that's not going to help us to identify it. Might be an anosogby, but we can't tell because it's too blurry. So definitely want that mushroom in focus, and you want that photo clear. So that's just a no-go off the bat. So the bottom picture, I said no, because if it was by itself, we would not be able to tell what that was. Those look like puffballs to me, but they're not. They're agaricus field mushrooms. And so the ... is just to say, if you have this photo as an additional photo to other pictures of the gills and the cap, then that's great. Then we can see the habitat. It's in a grassy field. That's wonderful. It's good. I always try to get an out shot if I can. So this is helpful if it's an addition, if it's paired with other photos. And then the one on the top here, a lot of people will take pictures of a mushroom from the top, and it's just like a white circle, and you know, you can't ID a mushroom like that. And this picture is actually pretty cool because you can see the texture pretty good of the cap. And so once again, in addition to other photos, pictures of the gills, pictures of the stipe, the habitat, if possible. And so I kind of thought that this picture reminded me of the moon. So, you know, I could be looking at, I have bad vision, so I'm squinting. I'm like, is that the moon? So had fun with that one. All right, back to the words. All right, so collection and processing. This is, you know, what you're gonna do if you find a great mushroom and you wanna bring it home to ID it or to dehydrate it for specimen purposes for the sequencing. So I use a tackle box, and some people like to use wax bags and they keep them in their basket, but you can keep a tackle box in your basket too, and they're much better and less likely to squish your mushrooms, and you can get them a variety of places, hardware stores, craft stores, what are they called? Thrift stores have them a lot, and dollar store sometimes has them. And they're just a great way to, you know, you're out there, you're moving around, it keeps your mushroom intact because you're way less likely to, you know, be able to take it home and look at it again or dehydrate it properly. Because if you have torn up pieces of a bunch of mushrooms, then you could very easily send the wrong one in for sequencing. So you want to keep them separate from each other, because that's the thing, is you're trying to follow this process through from finding it in the field to getting it sequenced. So we want to know that the mushroom that you have dried is the one that you photographed and the one that we are hoping to look at. As you know, you could be really easy, everyone could be disappointed this way if not. So yeah, and they look very different dried too. So alrighty, and use a dehydrator or the proof setting on the oven to dry it. If you know, a lot of people don't have dehydrators, I recommend getting one if you want to either dry mushrooms for culinary purposes or for specimen purposes. And you want to keep it below 155 degrees as to not degrade the DNA. I thought it was way less. I keep my dehydrator on the line, but I light. And you want to make sure the mushroom is fully dry to a crisp. You know, obviously now it's going to shatter depending, but if it has any moisture in it, it could mold. And I made a mistake of, you know, drying mushrooms not completely, and then I would store them, or I would dry it completely and store it in a brown bag. And if moisture gets in, it's going to rot that mushroom and it's not going to be viable for sequencing. So
Ness says the maximum temp is 100 degrees.
I had a feeling Steve was going to say something. So Steve, Sigrid commented this on, that's why I put it on here. I was kind of shocked. And there's a paper that says that 170 Fahrenheit is the most to keep the DNA intact. And so I would definitely say, I thought it was less, I would go with less, but if, you know, Sigrid is someone who's taken control kind of the new iteration of the Fungal Diversity Survey. And so I trust what she said. She wrote this on the little blue polypore observation, which is on the rare list. So definitely go lower than that if you can, if you want to be paranoid and safe, and you can do your research if you need. But like I said, if your dehydrator has a lower setting or your oven, it's going to still dry. So clearly we're worried about it. All right, so label your specimens when you are dehydrating them. See, this is from the old mycoflora project where you would have a protocol tag and you could cut out this bottom piece and place it in the dehydrator with the mushrooms that you knew it was sticking with that one that would go with the rest of the tag with the name and details. And so since we don't have that as part of the protocol anymore, you can just do your, to help yourself keep them sorted out if you have a lot of specimens. If you have just one, you're going to know that's the one that you put in there. But if you have separate ones and they are kind of similar, I would recommend either taking a little piece of paper, post it, write the name of it, or more specifically, write the iNat number so that you can follow it through. You know that because you could pick 3 different collections of the same species as well, and you don't want to get those mixed up. So you can take a little paper, you can put it in the dehydrator, and then you can pop it in a little at the plastic bag you store it in, so that when you hand it off to us, we make sure we know, because we need that iNat number. And the way to get it is that it's just in the URL of your observation. So go to your observation, it says observation, and then there's a slash, and then there's kind of like a 6 or 7 digit number. That's your number. If you need to see it after, I can show you. And then if you don't do it at that point, when you hand us the specimen, we need it. And I write mine on the plastic bag that I store them in, which I will show you. And yeah, you can keep them in a glass jar as well if you don't want them to be in the bag, but you want to just keep moisture out. So. And then here mine are, I just took this picture, some of my specimens, and I have them in sealed bags. I have little bags and big bags. And I put grains of rice in there just to keep it, as a desiccant, but you don't need to do that. Since I've had specimens go moldy, I got worried about it. And Steve thinks the rice is funny, so. All right, so what do I do if I find something I want to contribute to a project? So here's the steps. You collect it with the way that, just went over. Dehydrate it, store your specimen, because it might not be, we can't immediately get it from you. It might take a few months before we can actually send it out. So in the meantime, store it safely. I keep mine in a little box, have them labeled so you know which one's which, because even if you think you're going to remember, you're probably not. So keep it organized. And upload your photos to iNaturalist and enter it into the project page. And then email Steve for now until we figure out anything else, which this is the vice president at salsoundmushroomclub.org. And so you have to be a member to submit something in for sequencing, just so you know. And the amount of times we send in batches varies. It takes us a few months to do the whole process. It's a lot of steps. And it's a lot of work for Steve to kind of sort it all out, get all the specimens, make all these spreadsheets and stuff. And then it takes a few months to get usually, which ideally would be less, but typically a few months to get the results. And they're hoping to make that time frame shorter with the new management of the project. And the price can also vary. And so sometimes we get grants pretty often where they're covered for free and you won't have to pay, but that's not always the case. The club is really generous and can pay half of the cost, but that's to a limited amount as well. So you can't just have them, send 20 in. But you can talk to us about that. Some of these factors change, like I said, and the timing definitely changes. And it's not super expensive. I think the total price ranges between 15 and upwards of 27, depending on if we send it somewhere private, and in which case then the club might pay half, depending. So it's as affordable as it can be right now, because they're not just sequencing it, which requires expensive machinery in itself. They're also doing the DNA extraction, the PCR, and the sequencing. So, and that's labor and etc. All right, I'm almost done. So here is the virtual iNat ID clinic, which is inspired from the Wisconsin club's structure, and I didn't get to attend it, so I might need some help, you guys who are there. I think we got Rose and Leah who know about this. So I tried my best to just set this up in a way that makes sense, and it's going to be on Sunday, February 7th, and there's a Google sheet that I put for a sign up, and it's in the newsletter. I can pop it in the chat when I'm done with this. And it is for members only, so please don't sign up if you're not a member, but membership is super affordable and awesome. I recommend it, even if you're not in the area, especially now because we have great virtual talks. And so, yeah, upload. I just made a project page for this event. And if you type in SSMCID clinic, you can join it, and I can send that link also. And you can, so what we're going to try to do is that you can upload new findings and new observations, add them to the project, or you can put any degree of old observations you have into the project. And the goal is for us to just, me and Luca are gonna kind of help with ID, and whoever's present, we're gonna pull up their observations and kind of walk through ones that they're not sure about, or ones that they're like, they got an ID, but they're not positive or whatnot. And so it's kind of gonna be a new thing that we've never done. And so, you know, yeah, please come if you're free, and just, it's gonna be a very loose format. So just be ready to talk about mushrooms. And that's it. Thank you so much for coming today. Here's my email and my iNat username. So you can follow people on iNat too, part of why it's kind of social media-esque. And I just added this underscore to separate my last name because it's so short. And so I have my Instagram up there too. And yeah, I'll leave this up for a second before I exit out. But yeah, that's it. So thank you guys for sticking through this. Are there any questions?
You're getting some nice thank yous.
Oh, let me exit this. I'm going to exit this, guys. And I guess I'll just follow up by saying that, you know, we do this because, oh no, wait a second.
No, don't leave the meeting.
No, stop share. All right. Yeah, we do this because we love to learn. You know, it's not like science can be scary and intimidating, especially if you're not in the field. And I'm kind of in the middle ground where I've only in the last few years been working towards my bachelor's of science, but I learned a lot of this before I got into school, and I felt intimidated by, you know, these people who've been doing this for a while, and I was just felt like inadequate. But this is a way for you to kind of take your learning into your own hands, and using this really great resource of iNaturalist and also the Fungal Diversity Survey, you know, to connect with people, especially right now. It's really cool to use it during the pandemic and get out and connect with people. But even before that, just to meet